Before I begin, I'd like to thank Matt and Andrea for the invitation to provide this update for you on coffee germplasm and coffee leaf rust resistant varieties that we're working on here at the Daniel Kainoe Pacific Basin Agricultural Research Center here in Hilo, Hawaii. And so just as a reminder or refresher, we are a part of the National Plant Germplasm System. And so we are one of many plant repositories located throughout the country, responsible for important agricultural crops. And so we're one of the three main crops or three main repositories for subtropical and tropical crops, and we're located in Hilo. There's also a repository in Miami, Florida, and Mayaguez, Puerto Rico. And so if you notice, Miami, Mayaguez, and Hilo, we're all in hurricane-prone areas. And so we work well together to back up each other's collections. And so as a whole, the NPGS conserves germplasm from over 16,000 plant species. We distribute over 200,000 items for research each year. And so our primary crops at the Hilo repository include pineapple, breadfruit, starfruit, peach palm, pili nuts, and papaya and relatives. We were fortunate to be able to add coffee to our germplasm list thanks to your stakeholder support and congressional support as well. And we also have longan, lychee, macadamia, acerola, rambutan, and pulasan, and passion fruit and guava in our collection. Okay, so our coffee germplasm, as I mentioned, was started in 2019, or we were able to get funding in 2019. The funding also provided genotyping assistance with our colleagues in ARS Beltsville. We were able to provide some infrastructure for the importation and quarantine of new germplasm to be brought into Hilo, Hawaii. Our live backup collection is located in ARS Mayaguez, and we're also looking at long-term cryopreservation or deep freezing of the coffee material with our colleagues in ARS at Fort Collins. Okay, so for the genetic characterization of our coffee germplasm, we're very fortunate to work with Dr. Dapeng Zhang, who has developed this genetic marker panel using something called a SNP, which is a single nucleotide polymorphism. Basically, that just means that there's a base change that occurs within the population so that we can actually distinguish different sets of coffee from each other. So that way we know if a coffee has more Ethiopian in the background, land races in the background, or a Caniflora or some of the Timor hybrids. And so this has been a very, very useful tool in helping us characterize our germplasm. So our initial germplasm, because of the importation challenges, we were very, very fortunate to work with Dr. Chifumi Nagai from HARC, or the Hawaii Agriculture Research Center on Oahu. And so she had the foresight to collect different coffee germplasm, and looking forward, she actually was able to breed for a little bit for the coffee leaf rust resistance. And so having that valuable germplasm, we're able to have Dr. Dapeng Zhang actually genotype all of these to verify the collection and the identification of all the new germplasm coming into the repository. In addition, once we have germinated the seeds in Hilo, we planted them in the field, and we've also collected that material to be genotyped once again to make sure that there was no mislabeling or anything that we distribute will be verified. Okay, so now we're going to kind of switch gears to the varieties, looking at the different coffee leaf rust resistant varieties. And so if you remember back to Dr. Lisa Keith's presentation, she talks about the disease triangle. And so if you think about it, so for a disease to occur, we must have three conditions must be met. First, we must have a virulent pathogen, which in our case is Hemilia vestatrix, race 24. We have to have a favorable environment for the pathogen to germinate, and we also need a susceptible host, which unfortunately we have with our typicas, our bourbons, and our mochas. And so what we want to do is be able to prevent the disease from occurring. So what we can do is disrupt one of the conditions. And so Dr. Melissa Johnson has talked a little bit about her survey and monitoring to know when to do some of your field management, such as spraying fungicides. In addition, we know that pruning will help with the air circulation, and also good nutrition will improve the general resistance of the plant or tolerance of the plant to the disease. And our focus is actually looking at the race and cultivar interaction, or so looking at the bringing in different host plants of coffee to prevent this disease from occurring as well. Okay, so we do know we were very fortunate to have Dr. Vitor Vareza from University of Portugal to do the race determination for the coffee leaf rust that was found in Hawaii, and he determined that to be race 24. And so here you can see that there, for every resistance gene, which is in the second column here, you see that there is a virulence gene in the coffee leaf rust or in the fungi. And so what we want to do is we want to select for material that have the resistance to race 24 in the variety selection. And so one of the first things that we did is we looked at local coffee leaf rust resistant cultivars. And so these were basically when we saw massive defoliation of certain fields, we did see some plants within the field that did not have that severe defoliation, and the plants actually looked quite healthy under the pretty high disease pressure. And so here you can see a field managed by one of our local growers, and you can see the green coffee trees among the defoliated ones. And so basically we're able to identify some of this material. He named it after his children, so we have Juan, Cruz, and Maite. And so we were able to send some of these samples to Dr. Da-Peng Zhang, and we were able to determine that the parentage or the parents of these plants are from Ethiopian and Sachemore. So Sachemore does have that Timor hybrid, so it should have some resistance or tolerance. However, we do see a little bit of coffee leaf rust developing on Juan and Cruz. Although it is somewhat tolerant, you will see spots eventually. However, Maite seems to be holding up for resistance. So in preparation, in case these were important, so these lines were initiated into tissue culture by Steve Starnes, working with Dr. Mike Shintaku at the University of Hawaii at Hilo. Um, and then since Dr. Shintaku's retirement, some of the maite were sent or have been sent to HARC for further multiplications. So we do know that the maite does have the Ethiopian, Sarchmore, and Bourbon in its background. And the coffee leaf rust resistant was confirmed by Dr. Lisa Keith using leaf disc acid. Okay, so we want to do a special thank you to Chai from Greenwell Farms. They were able to cup the Maite coffee back in December of this year. The cups were floral, chocolate with a bright acidity and slight thin body. And this coffee is very geisha-like. And so because this is a promising cultivar, we are propagating these at HARC. And hopefully then we'll have material for growers to actually test this variety in their fields for yield and other qualities. So we know it's coffee leaf rust resistant as well as a good cupping coffee. So that's why this one has been moved on for further testing. So now I'd like to move on to the importation of other coffee leaf rust resistant varieties that we have imported either from international or I guess from the continental US. So just as a reminder, importation of coffee from international sources are actually prohibited coming into Hawaii or Puerto Rico. And so all coffee must be brought in through a controlled import permit through USDA-APHIS. Likewise, material from anywhere in the continental US to Hawaii is also subject to a quarantine from the Hawaii Department of Agriculture. So the International Multi-Location Trial, Variety Trial that we received from Florida. So this is a trial started by Row Coffee Research, and what they were able to do is ask all the countries in the world, what is your best variety? And what they were able to do is gather all of these varieties and then grow it in different important coffee growing regions. And so this is the first ever side-by-side comparison of the best of the coffee world. And so we were fortunate to work with Dr. Shifumi Nagai, and we were able to get in the tail end of this variety trial. And so we do have this material in our possession for evaluation. Okay. So in general, the IMLVT coffee varieties, so they list the varieties as being either susceptible or tolerant or resistant to coffee leaf rust. However, I just want to point out that this is in general what the resistance and tolerance is relative to the general Hemilia vastatrix and not necessarily race 24, because we know that things such as geisha is tolerant or resistant to race 24. However, it is susceptible to many other rust races. And so this is kind of a generality of the relative susceptibility and tolerance. And so in addition to the materials that are provided by the IMLVT trial, we are also using Typica or Katuai, depending on where these will be placed into the field for testing. This way we have our local control for any type of yield comparisons, taste comparisons, etc. Okay. So luckily, World Coffee Research was able to, because this study has been going on for a while. They do have global yield evaluations, and so we know that from their, in general, the F1 hybrids perform well in variety of climates. And so this includes EC16 and Central Americano, Ruru 11, which is a complex hybrid, and EC15, which is also called Mundo Manx. And so yield-wise, these performed really, really well in average over the different coffee growing regions. They're also able to do some cupping, and if you notice, everything has a score above 80, so it is considered a specialty coffee. However, we do see the higher ones, including the Marsalesa Batian, again EC16, which is the Mundo Maya, ranking in the top area of in the top scores. Okay, so we were fortunate that World Coffee Research was actually able to provide us with some of this material. So they provided us with about 100 grams of green beans, bulked from the different IMLVT coffee growing locations. We were able to use Kona coffee provided by Greenwell Farms as our local control. These were roasted before being shipped in to us in Hawaii by Gather Coffee in Texas, and it was cupped by local Hawaii Q graders at Pacific Coffee Research. So special thank you to Madeline and Brittany, and also Julie Burden, who actually flew down from Hark to do it. In addition, we did some additional cuppings where we had a grower panel, and also we sent some material to Chai at Greenwell Farms as well. And so some of this material, here's the coffee cupping group that participated at PCR. In addition, as I mentioned, we tried to get the most out of that 100 grams of green coffee. And so we did have a grower panel as well as our PBARC panel evaluate these cups as well. And so here you can see the results. And so here's the final cupping scores from the full cupping that occurred at Pacific Coffee Research. And so we can see that this is just the top 12 of those cupping trials. In addition, the top scoring varieties from the grower trial as well as from the trials at Greenwell are highlighted in red. And so you can see that there is definitely a location effect. So the lines grown in Nicaragua tend to score a lot higher than some of the other locations. But however, we do see varieties such as Batian, Mundo Maya, and Central Americano scoring well and repeating in the grower preference as well as the subsequent cupping at Greenwell. In addition, we were also given some coffees to cup in January, and so these were from Rwanda as well as the Democratic Republic of the Congo. And so these, except for the Marsalesa, did score above 80 to classify it as a specialty coffee. Again, the ones in red did score above 80 even after maybe about a month or so for the second set of cuppings. And so here we can see that the Batian also scored very well. And so what we're doing is the Hawaii Department of Ag has developed guidelines for the importation of new coffee varieties into Hawaii. And so we are working with the tissue culture labs in California and Florida. And basically, what we're able to do is, what we're attempting to do is bring in new varieties to Hawaii. And so just to back up a bit, so for the materials that came in on the IMLBT trial, these were obtained through an agreement with Royal Coffee Research, and for the purpose of doing research to see what performs well in different growing climates in Hawaii. However, we do not have the ability to do commercial production with these lines. However, World Coffee Research said that if we can get the breeders, or we can negotiate the breeders' rights with the people who develop these varieties, then what we're able to do is then grow them. Then they'll sign off on their contract, and we'll be able to use the material that's already in Hawaii for commercial production. And so, I guess extending beyond that, what we're able to do is we're working with Kimo Faulkner from the Hawaii Coffee Growers Association, and we've been meeting with the various breeders. So for Ecom, who had developed the H1 Centro Americano as well as the Mundo Maya, they were working with a lab in Florida, and so we're going to work with them to get the permit so that we can develop a protocol to safely import this material from their lab in Florida. And so the goal will be that part of the quarantine period can be done in tissue culture and then it can be kind of finished off here at PBARC. And so within the 1 year period, we're hoping that it can be inspected in Florida and Hawaii, so then it'll kind of shorten the time that it actually has to be in quarantine in the state. And so this is kind of a work in progress, and we'll keep everyone updated. Okay. So for our APHIS controlled import permit, we do have quarantine facilities. We recently purchased BSL2, which is a biological safety laboratory 2. It's a containerized lab. Basically, we could work with human pathogens in it, but basically the point is that it should be able to contain any potential pathogens that could come in with the imported material. So basically, we have an inspection scheduled with USDA-APHIS and HGOA actually this week. And so hopefully we'll be able to use this material as well to start our quarantine, or extend our quarantine capabilities. And so here's some photos of the laboratory. And so you can see that it is surrounded by a moat to prevent crawling insects from entering the laboratory. Within the lab itself, there is an anteroom. There's an entrance room, an anteroom. The anteroom has interlocking doors so that both doors cannot be opened at the same time. And then this enters into a culture storage room. And so we're hoping to get all the coffee materials grown on all these shelves with lighting. And so we're hoping to be able, right now, all of the material is in tissue culture. And so we would like to be able to start acclimatizing these plants from tissue culture into potting media in this laboratory. In addition, we do have other coffee varieties that we're bringing in. These are not necessarily in the IMLBT lines. However, um, they are supposed to have superior cupping quality as well as yield. And so these are brought in as tissue culture and will be under a 2-year quarantine. And hopefully then will become available. Okay. So as I mentioned earlier, we have we have been negotiating for different rights to coffee in the group, in the IMLVT. And one of them that we've shown interest in is Batian. And so this is one of the ones that has shown potential. And unlike the H1 varieties, or the F1 varieties, I'm sorry, Centro Americano and Mundo Maya, that need to be propagated by tissue culture, these Batian is something that could be grown as seedlings, and so this would be more similar to how coffee is produced in Hawaii now. So this would be similar to our Typicas, our Bourbons, our Mochas, where you could harvest the seed and it'll come true to type. And so we are in the process of trying to get the rights to Batian. And so this material was actually kindly provided by the researchers at Calro, the developers of Batian. And so this is Dr. Dane Cheserek and Dr. Elijah Jichuru. And so it is the advantage of the Batian is that it does cup well, it has pretty high production, and it does have resistance to coffee leaf rust, but in addition also coffee berry disease, which is a devastating disease in Africa. So this would be a good addition to our genotypes. Here you can see Batian growing next to Ruru 11 and SL28. And so you can see the yield on the 2-year-old Batian plants. Okay, and with that, I'd like to acknowledge all the people that have worked to help bring this to fruition. This is a great group effort, and I'm very fortunate to work with very talented individuals, and so their help is extremely appreciated. And also just to acknowledge our funding source. So part of this work is provided by the USDA NIFA SERI grants through SHAC and HARC as well. And with that, as they say, it takes a village, and so I'm just fortunate to work with a wonderful group of people and who are always game to take on any task, and for that I'm super appreciative. And with that, I can answer any questions.